So our next presenting company is Pluristem, and Pluristem is developing PLX cells, or placental expanded cells, which are derived from mesenchymal-like adherent stromal cells, and they're expanded in the company's proprietary 3D manufacturing platform. The company is developing PLX PAD in multiple indications, as well as PLX R18 for bone marrow transplant and also acute radiation syndrome. Here to present for Pluristem is Karine Kleinhaus, who's Vice President of North America. Hi, Karine. How are you?
Hi. Great to be here.
Thanks. So, Karine, maybe if you want, I can ask you some questions and we can kind of go through the slides. If it works that way, great.
All good.
So maybe you can start out by describing why the placenta itself is such a good source for cells.
So the placenta is really a fabulous source, at least in our opinion. It has a lot of things going for it. First of all, we get it at the time of delivery of a live, healthy baby. So it's really the youngest adult cell that you can get. So these are healthy, they expand easily, and they also have a lot of special properties. If you think about the placenta, it's the gateway between the mother and the baby as the baby's growing, and it has to work that way in two immune systems without being destroyed. And so the cells from the placenta are immune privileged by design. They're missing certain markers that we've shown and published that they modulate immune response. And so they are really the perfect source for allogeneic treatments. And we don't need to pre-treat. We've used different placentas or the same placenta into the same patient twice without an immune response. So that's really support for the fact that these are a truly allogeneic product.
That's really helpful. And just to kind of keep going along the sort of Pluristem approach, what's so important about this 3D manufacturing, and why is that really unique to what you guys are doing?
So the 3D is another, manufacturing is another important part of our company and a big differentiator. First of all, from a sort of cost of goods perspective, because we use the placenta and with our patented technology, and we just announced that we've got over 100 patents now, we can make 25,000 doses from a single placenta. And the reason we can do that is because this 3D technology allows the process to be more like it is in the body for multiplying cells or expanding cells, and that's why it's so efficient. The second thing is our factory and technology has been cleared by the FDA through phase 3, by Europe through commercialization, because we're starting a phase 3 trial and Europe needs it cleared through commercialization before you start the phase 3, and they recently, a few weeks ago, visited our site. So the factory is commercial ready, and that was a strategic decision so that we would have it ready without having to make adjustments, without having to change technology as we scale up. So when we expect or are hopeful that our phase 3 completes successfully, we're ready to produce for the market and have been certified that way.
Great, that's helpful. Now, the lead program is PLX PAD, and you're evaluating this in multiple indications, but as you mentioned, lead indication. So maybe you can tell us first about these cells and what makes them unique, and then maybe some of the data that's been generated to date.
Great. So we have two distinct products, and each one is in the clinic right now for two indications. Our lead product is PLX PAD, and what it does is address growing new vessels, proangiogenic, reducing significantly immune response, which is to reduce scarring, and regenerating, stimulates regeneration of muscle. So our cells, both our products work by paracrine endocrine effects. So we inject them in the muscle with a regular syringe. They sense cytokines or chemical signals from damaged tissue, and in response to that particular signals of cytokines, they secrete a range of therapeutic proteins. PLX PAD has its own range, which includes multiple, about 20 proteins, which target those functions.
Excellent. So it's really more of a paracrine effect that these cells are having?
No, it's actually endocrine. We treat with our second product bone marrow regeneration, and we still just inject it IM into the muscle of the leg. They really secrete and have a crosstalk with the damaged tissue. So it's clearly systemic effect.
And how often are these injections given? Do the cells thrive, and do they have a durability? What does that look like?
So we give them in different regimens depending on the indication. So for our lead indication, their phase 3, we give in something called critical limb ischemia, where you have blockage of blood flow to the leg, tissue breakdown, gangrene, amputation, and a high risk of death. In this case, when we're in our trial, we give 300 million cells at the start of the protocol, at the start of the treatment, and 8 weeks later, we give a boost of another 300 million cells. And then we follow them forward for a year or longer because on our the protocol for our trial. In the case of hip replacement or hip surgery, we showed phase 2 data, which was actually really nice and personally very exciting for me, which showed that if you inject the cells one time at the time of hip surgery, that was all we needed to get a really fantastic result 6 months out.
Great. So let's pick up and dig a little bit more into the critical limb ischemia indication. Maybe you can tell us about what you saw in phase 2 and what the effort is moving into phase 3.
So we did 2 phase 1/2 trials, one in the US and one in Germany. And what we did with them was determine a few things. One is that we want to boost. We want to have, for that particular indication, we want to have 2 injections. The second thing we determined is the character, the class of patient in that indication, and we chose for our phase 3, which is beginning now, Rutherford Category 5, which is not too far gone in the sense that the leg isn't, doesn't have too much gangrene, but we chose specifically patients who are not eligible for the vascular procedures that are standard of care in this group of patients.
Great. And tell us about the phase 3 trial design.
Great. So the phase 3, building on what we learned in those phase 1/2s, is starting now. We're recruiting in the US and in Europe for a 250-patient trial, which we are very excited to begin in about 40 sites. We were accepted into the European Adaptive Pathways Project, which gives us the right or the opportunity halfway through patient recruitment and data collection to have an interim data analysis and allow us to apply for conditional marketing approval at that time, which is a really fantastic opportunity. In terms of trial design, 300 million cells at first treatment, 8 weeks later, another 300 million cells, or placebo.
Okay, great. And what's the primary endpoint?
Primary endpoint is something called time to event, either time to death or time to amputation. Time to event is often used in cancer treatments and the like, and it's a really nice endpoint. The reason is you get a lot more data from each patient, which allowed us to have a nicely powered study in only 250 patients as a pivotal trial. The reason, though, that is that for a patient who comes in at the beginning of the trial, let's say the trial takes 2 years or 3 years or whatever it takes, that first patient or second patient are followed all the way through the trial until they either die or have a leg amputated. So you can get 2 or 3 years of data from one patient, while the minimum is the last patient in is going to have 12 months of follow-up.
And how long do you think it would be until we get data from that phase 3?
So we anticipate that it would take 2 years to recruit all 250 patients. However, and then the last patient in is going to have 12 months of follow-up, which brings us out to 3 years till data. However, because of the European adaptive pathways, which we got accepted to, we can, after 2 years, so after half the patients complete a minimum of 12 months of follow-up, we can have an interim analysis, and that would allow us to get data much sooner.
Great. That's really helpful, very informative. Any questions on PLX PAD? So you mentioned, too, the hip indication, and I do want to spend some time to talk about the other product.
Yeah.
But maybe tell us about sort of what the improvements are. What are these cells doing in that indication?
So we did phase 2 there, where we were able to show, sort of clinically speaking, when a patient had a surgery for hip replacement, and that surgery damaged, as part of the treatment, it's expected that a certain muscle is damaged during the procedure. We were able to show that with a single injection at the time of surgery, 6 months later, that specific muscle was about 500% stronger in those who received our cells versus those who received the placebo, and the p-value was 0.0067. And we showed that the muscle was 300% larger on MRI in those who received our cells versus placebo, and the p-value there was 0.004. And we've shown in preclinical trials and in biopsies during that trial that besides what we know that it does with angiogenesis, we were able to show reduced scarring, larger muscle fibers, and that those fibers were truly fibers and not fat in terms of larger diameter.
Excellent. So that may have applicability for other surgical indications as well. Really interesting. So I want to switch gears. I really don't know a lot about this product. So PLX8R18,
yes,
I believe. And you're using it for bone marrow transplant, but also an IND was recently cleared for hematopoietic stem cell transplant. So maybe tell us about how these cells are different and how you're using them.
So they've really been shown and certified as a different product from the PAD because they secrete a different range of therapeutic proteins whose point is to trigger the bone marrow to regenerate itself and be able to produce white cells, red cells, and platelets. So we have multiple hematologic indications that we're going for. The one that's furthest along and maybe FDA approved by next year is treatment for acute radiation syndrome, which is basically when you have high dose acute radiation, like after a dirty bomb, which we're hearing a lot about now, or a reactor meltdown, like at Fukushima, who's working with our cells, one of the ways you die essentially is that your bone marrow is destroyed, so you can't make white cells, you die of infection, you can't make red cells, you die of severe anemia and heart failure, or you can't make platelets and you bleed out, essentially. So the U.S., we did many trials. We showed that when you give high-dose radiation to small animals, about 100% of those who get our cells survive, 30% of the small animals who got placebo survived. The U.S. government was very interested. They did a whole range of small animal trials looking at efficacy and repeating that, showing mechanism of action piece by piece in very elegant studies. And they already completed a large animal trial. These types of indications are approved through animal rule. You don't do radiation experiments in people, obviously. And so they completed a large animal model in non-human primates where they looked for dose optimization, dose selection, efficacy, safety, et cetera. We're expecting that data in the next few weeks. I'm really hoping and confident or expecting that it will be good. And as soon as we get it, of course, we'll let everybody know. And that trial would be springboard to a pivotal trial, which we're hoping that the NIH will run as well. Because until now, all the work and the funding has been by the NIH, the NIAID within the NIH.
Great. And then ultimately is the commercial opportunity for that indication, government procurement, something along those lines?
So we're also right now running a phase 1 in the U.S. for incomplete engraftment following hematopoietic cell transplant, which means basically you replace bone marrow that's not working either with other bone marrow cells or cells from peripheral blood. That's done standardly, but the problem is that sometimes they don't work. So they sit there, they're not rejected as the GVHD that was required before, described before, but they don't make enough cells for the patient to be independent of transfusion. That's the indication we've started testing in. That's one hematologic indication. There are multiple others that we have also in the pipeline, but that's already recruiting. In terms of the stockpiling and procurement from the government, that would certainly be something that we would go for. If the data is good in the next few weeks, we will keep working with BARDA to work on a funding and for the pivotal and to stockpile. Just in terms of, it's very hard. There aren't that many data points. But we're expecting that we can make a good run of it with the stockpiling if the data is what we expect.
Great, excellent. Well, Karine, thank you so much for being with us.
Thank you.
All the best on developing these programs, and I will certainly follow and pay attention and see how it all turns out with you. Thanks so much.
Thank you so much.